Hello everyone. Good evening everyone. Hope I'm audible to you. Good evening. पारुल मैम का वीडियो अपलोड हो गया? So how many of you have watched the video of Parul? Parul मैम का वीडियो कितने लोगों ने देखा है? So that will also come in the parts. First part. I've seen it. Hi Kusum, how are you preparing? Going good? How was the video? Is it good? Is it helping? Hi Sarita. Hi Halok. Hi Bhavna. Akashri, Sakshi. So there is one correction to make. Someone has rightly noted it in the comment section. I was just going through the comments. Can you tell me like what is the correction? Can anyone type? Yes, like what was missing in the previous video? There was some mistake in the previous video. A minor mistake. Don't worry, nothing is. Somebody catched it. What's the mistake? There was some mistake. Some part of explanation in the previous video. How many of you have seen it? Quick. Remember here I was discussing about the mini satellites and the micro satellites. So here there is one addition you can make. The example for the mini satellites is VNTR, okay, which are present in, where are these repeats are present? They are present in mostly the telomere or the centromeric region. Okay, the mini satellites, which are mostly used for DNA fingerprinting. Next, the microsatellites is simple short terminal repeats, STR, not VNTR here. It's just the STR, which are much more less in the size in terms of sequences, which are dispersed throughout the chromosomes. Please make this note correction. I'll update anyhow this in the PDF. Okay. Hope it is clear to everyone. So now let's continue our discussion where we left. So where did we leave in the previous video? And no one commented regarding this termination of eukaryotic replication. Why so? So after completing this video, comment on the video regarding the termination of the eukaryotes, how exactly the termination takes place. Fine. So let's start. So now till now we have covered the genetic material. We got to know about the replication and we learned a few things about the repair. And mostly we learned about the SOS response and the genes which are associated with this response, which are mainly two different types, two genes, lex A and rec A protein, right? The gene product of these two. So we came to know that this is the transcription repressor of the genes, which are the genes SOS genes, and this will help in cleavage of lex A from the SOS genes and makes it active. So is what we learned in the previous. Okay, voice is clear. Just check your internet connection. Voice is clear, I guess. I'm also seeing the frequency here. Okay, next thing is gene action. Let's start with the gene action. In the simple words, like gene action in the terms, see, when the molecular genetics was introduced, so starting with the genetic material, starting next with the structure, next with the replication, everything was devised in an orderly fashion. The next particular thing which was needed to be devised, it is regarding its mechanism or the metabolic pathway. How exactly it is responsible for the trait. We know that genotype is the, like, genes are the one which are responsible for the phenotype, but how it is? So for explaining this, there were several different hypotheses that came into existence. The first one was by the Gerard. It was in 1909, which said that one gene will produce the one metabolic block. That means if there is a pathway, A gives B, B gives C, and C gives D, and D is our final product. The block, if there is any block, the gene sequences or in the pathway, that will not result into the phenotype. So that's what the one gene, one metabolic block represents. So that was given by Garrod. Next, it was synthesized, improved, and then later it was realized that the one gene actually gives the one enzyme which can act on a certain metabolic pathway. So here there will be one enzyme, let's take A, there will be another enzyme B and C. So every gene will give us the one enzyme that was given by whom? This hypothesis was given by Beadle and Taunton. It was in 1941. Next, after that, it was again improvised and told that no, it's not just the one enzyme. The one gene that do not just give the one enzyme, it will actually give one polypeptide. So now tell me, one gene, one polypeptide. So one polypeptide represents one enzyme, or it represents just the polypeptide, where like multiple polypeptides have to be clubbed to form the enzymes. Which one is true? Thing is, even the one single polypeptide can be the enzyme, or the multiple polypeptides from the different gene products can club or aggregate later to form the functional enzyme. Both is true. So that's the reason it was even synthesized to form the, meaning that one gene actually gives the one polypeptide, not always the complete enzyme. That means the gene will not give us the complete enzyme every time. It will actually synthesize one polypeptide, which is a chain of amino acids. And depending on the enzyme, a single polypeptide can account for an enzyme, single enzyme, or multiple polypeptides can form the enzyme. And it was given by Ingram in 1957. So later, based on our current understanding, later based on our various research in the bio biochemical field, so it was finally known that okay, not always the gene will give the protein. Is it true that gene will always give the protein? Tell me. Whenever gene is transcribed, gene will always give us the enzyme or a protein. Is it true? Protein product? Yes. So based on our current understanding, we also know that yeah, enzyme is also true. Like one gene can give the enzyme. One gene also can give the one polypeptide, just a polypeptide. And the enzyme, one gene can give the RNAs, different types of RNAs, which may be converted, which may be converted to protein, or it can as a have the regulatory role by itself. RNAs by itself can have the regulatory role, or the RNAs which are synthesized can be processed or translated into protein. Both are possible. So that's why it was more simplified, and finally the answer was one gene gives the one diffusible product. So that is what currently accepted is. So this is everything about the biochemical path. So next, what you have to understand and what you have to remember regarding the amino acid, it is about the different categories. What are the different categories? You have to know the list of non-polar aliphatic, aromatic examples, the positively charged, the negatively charged, and polar uncharged. So you have also the acidic amino acids and the basic amino acid. the sulfur containing amino acids. So all of these are important for you. Just make sure that you know the amino acids. What is the total number of essential amino acids we have, that we have to remember? Yes, there are like n number of amino acids, but how many are essential? What is the exact number? Is the voice low for everyone or only for few? Yes, it is 220. So that's what you have to remember for your exam. Okay, categorize them based on like whatever I've done plus the acidic, basic, sulfur containing amino acids. So these are important for you people. Next, what about protein? So what is exactly the protein? Protein, if you know, like it is a stretch of amino acid sequence, right? So that's the polypeptide, and later it is packed into the protein. So now there are again four different stages of packaging. So primary structure, secondary structure, tertiary and quaternary. So this is like just like similar to that of chromosomal or DNA packaging. Here the first level of organization is primary. Primary is what? It is just the simple stretch of amino acids which are present linearly. Next, this is again condensed to form two different types of complex or the structure, which is alpha helix and the beta pleated sheets. And again this is condensed to form a tertiary structure, and lastly we have the quaternary structure of the protein which is present. This is the different stages of packaging that takes place in the protein packaging. That's it. Okay, now there is one concept called as collinearity of the polynucleotides and polypeptides. What is it? What is exactly the collinearity means? The collinearity means, like, if I say that this is like AT, GC, AT, GC. So this is TA, TG, A. Just a hypothetical example. If this is a gene, now whenever it goes for the mRNA, one of the strands will be transcribed. One of the strands will be transcribed, and instead of T, there will be in the mRNA there will be U. So this U, this mRNA is giving what? In the next step through the translation, it is giving the stretch of amino acids. The stretch of amino acid that is being given by the certain gene. Here it was noted that this sequence, the sequence that was present in the mature mRNA, the exact sequence it is present in the amino acids in terms of codons. The order was precise. There was no interchange in the order. That's what the collinearity means. And it was given by whom? It was given by the Crick in the 1956. The understanding that whatever sequence we have in the nucleotide, the same sequence will come to the mRNA. Once they are processed, the matured mRNA, after removing the introns, whatever the sequence that is present in the mRNA, the same sequence will be visible or will be formed in case of protein or in the amino acid chain. That's about the collinearity. You have to understand this. Next, let's talk about the step of transcription. Transcription is a process of conversion or converting the information or getting the information from DNA to RNA. So now mostly the thing is, like, we know it is mRNA. Is it true that having the information transcribed from the DNA, it is mRNA? That is what the transcription means, or all the conversion that takes place to RNA from the DNA, the generalized term is transcription. Which one is true? Is it mRNA or RNA? Just type it whether it is just mRNA or it is overall for the RNA, that we say that is transcription. Although like in the books it is mentioned that it's mRNA, but for the clear understanding, when the information is transcribed from the DNA to RNA, the process we call it as transcription. And this transcription takes place in which direction? This takes place in 5' to 3' direction. And with the help of what? The help of enzymes, we call it as RNA polymerases. These are the enzymes which transcribe the information from the DNA, where we know that DNA has double stranded. From the double stranded, one strand will be the transcribing strand and the other one is non-transcribing strand. So now tell me, transcribing strand is the coding strand or non-transcribing strand is the coding strand? One is the coding strand. Is it like the one which is getting transcribed or the one which is not getting transcribed, which we call it as coding strand? Yes, transcribing is coding. Do not get confused here. See, here if it is ATGC, here it is like TACG. Now if I say that this is transcribing, where in place of A it will be U, in place of T it will be A, and it is CG. Now this information is almost similar to that of non-transcribed strand. That's why this is called as coding strand, where exactly the information present in this strand, it is coded into the mRNA. It is like non-transcribing strand, which will be called as the coding strand. Okay, fine, good. So this is about the transcription. Now for our understanding, the first time the existence of mRNA, the mRNA was given by whom? It was by Spiegelman. It was in 1967. Try to remember the name of the scientist which I am mentioning here. And next, the RNA polymerase activity. This can come in the exam. The RNA polymerase, which is the major enzyme in the conversion of the DNA to RNA, it was discovered by Wyss in 1960. And next, the concept of flow of information, where the information flows from the DNA to RNA to protein, which we call it as central dogma of biology. Who gave this? It was given by, again, Crick. Okay, Colinearity was also given by Crick, and this central dogma was also given by Crick. Remember these points. Okay, next, this is fine, but now this is not the universal, right? Also know, like, this is not universal. If it is asked in the exam, you have to answer this whether it is in the assertion reasoning or in the statement wise questions. And because in some viruses we see that the RNA will be converted into DNA, which is cDNA, and then to RNA and the protein. Here the sequence is changing, so that's why it is not universal. Okay, now let's come to the process of actual transcription. So let us again, the comparison starts with the prokaryotes and eukaryotes. First thing, the prokaryotic transcription is polycystonic and the eukaryotic transcription is monocystonic. What is polycystonic? So the single start of transcription will have a genes for more than one generally. That means once the RNA polymerase command binds here, so it will transcribe the information for the genes which are more than one. This is called as polycystonic. But in case of eukaryotes, every start of transcription start site will only provide the information for a single gene. Okay, that is what the monocystonic means. But simultaneously, in case of eukaryotes, the many genes can be transcribed at a once, a single RNA polymerase in a single process. And I told you the direction of this and the direction of this transcription, it is 5 prime to 3 prime. It is for both prokaryotes and eukaryotes. Next, and this process is primer independent. If you know, like in case of replication, like what we required, we needed the primer to start the process of replication, and only then once the process is started, the DNA polymerase can extend the stretch where the primer is the must requirement in case of replication. But when it comes to the RNA polymerase, so this when it comes to the transcription, the primer independent process where the primer requirement is not there, both in prokaryotes and eukaryotes. But tell me whether this is template dependent or template independent process. Whether the transcription is template dependent or template independent. Do not get confused with the terms. One term was regarding the primer independent or primer dependent, where the process is primer independent. But whether this entire process of transcription is dependent or independent. How come it is independent, Ritesh? Like, that's why, like, I know, like, this is where with the terms you will be confused. You know the things, but still you will be confused. See, without template, how the mRNA is produced? The information in the mRNA is coming from one of the two strands of DNA, right? So remember, it is primer independent, but it is always the template dependent, both prokaryotes and eukaryotes. And coming into the RNA polymerase, how many RNA polymerase are present in the prokaryotes? How many? So there is only one, that is single RNA polymerase, which is present in the prokaryotes, which is as a holoenzyme consisting of the different subunits, where like alpha, beta, beta dash, omega, and sigma. So these four are called as core enzyme, plus there is a sigma factor. Okay, sigma factor. This forms a complete holoenzymes which is functional. But in case of eukaryotes, we have 5 different types of RNA polymerase. Can you list them out? So it is RNA pol 1, 2, 3, 4, and 5. And if you ask like what is the function of all of these, the first one, RNA polymerase 1, it is like rRNA and some of the SNO RNA, small nuclear RNA. RNA polymerase 2, what is its function? It is majorly for mRNA, and RNA polymerase 3, it is for tRNA. But the thing is, most of the rRNA, it is not RNA, it is rRNA. Okay, so also some sort of confusion everywhere. Is rRNA. Now here, along with the tRNA, it also produces one type of rRNA which is not produced by the RNA polymerase 1. Which is it? It is 5S. Remember, it is 5S rRNA. Remember, it is 5S rRNA. Okay, so this is about the eukaryotic RNA polymerase. Now, same thing, I try to explain it here. So one thing we got to know about like the enzyme. Next, it's about direction. Let's talk about the termination again. Since there is exact difference between the prokaryotes and eukaryotes, I have listed it out. So termination in case of bacteria, it has a two different strategies. So this is like intrinsic termination, and second one is row dependent termination. Someone in the previous class mentioned termination of replication. Someone used the term as row dependent. See, that's where we will be mostly confused with. See, this is related to what? This is related to the transcription, not the replication. Remember, prokaryotic prokaryotic replication prokaryotic replication termination takes place by the help of TUS gene product. Okay, here in case of transcription, the termination, it is because of intrinsic termination or row dependent termination, where row dependent is, row is a protein which will help in the terminating the process. Intrinsic termination, because of the structural changes within the growing chain, there will be a termination. And in case of eukaryotic, say prokaryotic termination is simple, because there was only single RNA polymerase which was responsible for the production of RNA. But in case of eukaryotes, since we have like different types of RNA polymerase have a different process. So that, for example, RNA polymerase 1, it is similar to row dependent. RNA polymerase 3, it is because of the polyuracyl rich signals that are present. And the RNA polymerase 2, it is like most complex among all of this, which is explained by two different models. Again, we have two different models here. See, these models you should never confuse, because earlier for the primer removal, if you remember the models, again we had two different models. So what are the two different models that came in place while we were discussing about the replication? Quick, so what were the two different models you remember? The primer removal models, like I told you in the eukaryotes. Yes, it is flap model and RNase model. Very good. It is the RNase model and flap model. What about prokaryotes? How does the primer removal takes place? The primer removal in case of prokaryotes, it is by the DNA polymerase 1, because it has which activity? 5 prime to 3 prime polymerase, sorry, exonuclease activity. I hope like you won't be confused with the exonuclease and polymerase. Polymerase is addition, exonuclease is deletion. Remember like that. It is by 5 prime to 3 prime exonuclease activity. Similarly, again we came up with the two different models, but this model, it is related to the termination in case of eukaryotes. One is allosteric model and second one is torpedo model. So you can just have one look at it and you have to remember these models. That's important for you. Now, enzymes for the transcription. I told you about the RNA polymerase. So it was discovered. Who discovered it? I told you the name, Viz, and it was Gerard Artvis. Like most pronounced name is like Viz. Remember the Viz at least. And if you can remember the Artvis also, please do it. In case of prokaryotic, it is single type of RNA. Most of the things I already explained, and it has a holo enzyme. Please remember the holo enzyme subunit, plus remember the category that this is called as core enzyme, and the last part is like sigma factor. That's very, very important. Plus, what are the activities that you have? It doesn't have any exonuclease activity, no primer is required, and 2 phosphate groups are consumed per nucleotide is added. Whenever the growing chain in the mRNA, the nucleotides added, 2 phosphates groups are consumed externally. So that's what you have to remember. Plus, the functions of all of the subunits is given clearly here, starting with the alpha subunit. So this is required for the assembly, and the beta and beta dash subunit, it is actually having a catalytic center which is involved in the chain elongation, and the omega subunit, it will facilitate the assembly of the enzyme which is the RNA polymerase enzyme, and the sigma factor, it is concerned with the promoter recognition, where promoter recognition means there will be a stretch of sequences in the DNA where the RNA polymerase can go and bind. So these sequences are called as promoter sequences, which have the binding regions for the RNAs, and this is helped by the sigma subunit. Okay. So now there is one more type. This is prokaryotic RNA polymerase. But there is one more type of RNA polymerase which is RNA dependent RNA polymerase. So this RNA polymerase which we talked about, it is DNA dependent or RNA dependent? DNA or RNA, which we talked about this here, the enzymes for the transcription. Prokaryotic RNA polymerase is DNA dependent or the RNA dependent? Just quick, this polymerase which we are talking about, which we discussed in detail about the subunits and everything, it is, remember, DNA dependent. Why it is DNA dependent? Because it is transcribing the information from which strand? DNA strand. Again, we also have like RNA dependent RNA polymerase. Why it is required? It is required because viruses do not have the DNA. From the RNA directly the information has to be transcribed, and this information will give you the protein for its survival. This is how the process takes place. That's what we have seen in the central dogma also, right? There are two different types. Viruses can directly go from RNA to RNA to protein, first part. Next part, like we saw that RNA can first go into the DNA, and DNA can next go into the RNA, and next to the protein, right? So if second is the case, first is the case, then it will be by the enzyme, the RNA dependent RNA polymerase, and it will, again, the direction won't change. If they try to confuse you in the exam, that DNA dependent RNA polymerase works on 5 prime to 3 prime direction, but the RNA dependent RNA polymerase works on 3 prime to 5 prime. Seeing the difference in the terms, do not get confused. It's universal that the transcription takes place on from 5 prime to 3 prime direction. There is no change in it. Even the transcription, even the replication, both starts with the 5 prime to 3 prime only. And the eukaryotic RNA polymerase, I told you about the 1, 2, 3, 4 and 5. The 4 and 5, there is no prominent importance to this. So this has been thought to be evolved from RNA polymerase 2, and it is required for the biogenesis of the siRNA. What is siRNA? siRNA and miRNA. You have two different types of RNA. MicroRNA and what is siRNA? Small interferon RNA. Okay. So this is about the RNA polymerase 4 and 5. Next, apart from this was all about the nuclear RNA polymerases. Why nuclear RNA polymerases? I am saying it because in the eukaryotic cell, the DNA is present in which of the parts? One, it will be in the nucleus, and other parts can be also present in the mitochondria and in the chloroplast, right? So this should be also transcribed, and the protein synthesis has to take place. So here, that is because of the organellar RNA polymerase. So let's see like how it is different in case of mitochondria and the chloroplast. Remember, the mitochondrial DNA polymerase, it is encoded by the nuclear genes, and this RNA polymerase, how it is formed? It is formed by the nuclear genes. And the chloroplast, there are two types of RNA polymerase that is found in the chloroplast. Again, the single organelles have the two different types of RNA polymerase. First one is chloroplast encoded, and second one is nuclear encoded. Nuclear encoded is same as like mitochondrial, but again there is one more thing that is called as chloroplast encoded. That means the RNA polymerase is itself encoded by the genes that are present in the chloroplast, which is similar to the eubacterial type or the bacterial prokaryotic type, again which has same similar subunits. This is what you have to remember regarding the organellar RNA polymerase, which is different from the eukaryotic RNA polymerase. Okay. So in the exam, if they ask you like mitochondrial transcription takes place by which enzymes, it is not by the RNA polymerase 1, 2, 3, 4, 5. It has its separate RNA polymerase, and when it's come to the chloroplast, it has two different types. One is encoded by chloroplast itself, and the other one is encoded by the nucleus. That's what you have to remember. So what is the next step? Now tell me, like once the DNA is synthesized, sorry, RNA is synthesized from the DNA, what is the next process? So let's assume that there are three different types. Let's talk about only three different types. There are also the other types of RNA. So this is in case of prokaryotes or eukaryotes, which I am talking about, the processing. The RNA processing prominently takes place in prokaryotes or eukaryotes, or it is both. What do you think? So remember, the prokaryotes will almost do not undergo any processing, where that process of transcription and translation, the process of transcription in translation, it is like continuous. There is no as such gap between them, and everything is taking place within the single nucleosome. Okay, what is nuclear membrane which is formed within the bacteria? Within that, the continuous process of transcription and translation takes place. There is no as such gap, and there is no RNA processing or processing of RNA that takes place in case of prokaryotes. Just that you have to remember. And the whatever we speak about the RNA processing, it is only related to which? It is only related to the eukaryotes. See, such type of questions can definitely come in the statement wise, that the RNA processing, the prokaryotes have the RNA processing, the eukaryotes undergo the RNA processing, extensive processing, any kind of questions related to this. So you have to be very sure that the prokaryotes do not undergo any processing. And in the eukaryotes, is it the mRNA which undergoes the processing? RNA processing is only concerned with the mRNA, or it is for all the other types of RNA too? What do you think? It is mRNA for all? All are mRNA. Just type in the chat box. All. Okay, all. Yeah, that's what. Like you have to remember that it is not only the mRNA. All the other types of RNA will also go for the extensive processing. But for us, like for our understanding, what we do always, we only study the mRNA, since it is the one majorly producing the protein. Okay, that doesn't mean that these two doesn't go processing. This also go for the extensive processing. But what we study and what is important for us, it is the processing of the mRNA. Now tell me, in eukaryotes, mRNA processing, what is the steps of mRNA processing? Sequence of steps. So it is starts with 5' capping and goes with the splicing and 3' polyadenylation, or it is commonly called as what? 3' capping and 5' capping and 3' tailing, and next there is a last process which is called as RNA editing. But there is some problem here. The problem is, although like in the books we study that all the 4 steps are related to the RNA processing, but in one of the previous year exam, in the key answer, they did not mention the RNA editing to be the part of RNA processing, because not all RNA will undergo editing. Because of that reason, might be the case that they have removed this. So remember, these three are the prominent part of the RNA processing. So according to the ICAR, whenever the paper comes, this is what they have interpreted, and the key answer would be such that it will only have these three. Please remember, do not include the RNA editing. Since I have said that RNA editing is there, like do not please mark also include the RNA editing in the RNA processing, but it is the process which is done during the processing of RNA. Now, is the sequence correct? Whatever I have shown here, the sequence is like 5' first, and splicing second, and third it is 3' tailing. Is it right? Is it true? So this order of sequences, it's not always true that it is like overlapping. The process, see, whenever the DNA is present and mRNA is coming out, immediately once it comes out, the splicing can immediately occur, or 5 prime capping can also immediately occur, and once the tail is formed, the 3 prime tailing can will also occur. So they are like overlapping process. So there is no exact order of sequences. So if there is something which is asked in the exams, know that 5 prime capping is like primary one, but there is no exact sequence for it. Okay, so what happens in the 5 prime capping? What exactly happens in the 5 prime capping? The 5 prime capping, there is a G cap which is added to the whole stretch of, this is the mRNA. Do not get confused with the structure, don't feel it complex. So this is like stretch of the strand, the mRNA strand. To the last strand, you can see to the fifth 5 prime to 5 prime, there is the inverted phosphodiester bond which is formed, and this is, what is this nitrogen base? It is guanine. Okay, this is what the 5 prime capping means, where the guanine is added again as a 5 prime triphosphate bridge, which will lock the entire mRNA sequence. Why this locking is important? To bring the stability to the edge. Like ends will always prone to the endonucleases and exonucleases. If the ends are open, even the exonucleases can act on this and they can digest the mRNA to make it stable. Yes, it is 7 methyl guanosine residues are added at the end, which is called as 5 prime capping. Good. And here it is in the last case tailing. Tailing is nothing but adding of, like it is just the polyadenylation process where the series of adenine are being added to this 3 prime end, and this takes place at 5 prime end and tailing occurs at 3 prime end. So this is about the priming and capping and tailing. What is its role? I mentioned the role, protection of mRNA, and this is required to guide the transfer of mRNA from the nucleus to cytoplasm. I did not talk about this. See, whenever the mRNA is being synthesized in the nucleus, you know, like it should be going to cytoplasm, right? Where the actual process of translation takes place. Where exactly in the cytoplasm? It is in the endoplasmic reticulum associated with the ribosomes, rough endoplasmic reticulum. This is where the translation takes place within the cytoplasm. So for this transport from the nucleus to the cytoplasm, this capping will guide this transfer process. Okay, and this also have some role in binding of ribosomes with the mRNA. The recognition of recognition of mRNA by the ribosomes, which will also help by this 7 methyl guanosine is added to the 5 prime end of the mRNA, which has been freshly synthesized. Okay, this is about the polyadenylation. So next, what is about this splicing? Splicing is nothing but, in a simple words, it is nothing but removal exons and joining of introns. Removal of introns and joining of exons. That's what we know. But this is again some kind of complex process, and it generally consists of 2 steps. At least know this, that this is a 2-step trans-esterification reaction, and there will be a formation of lariat structure. There will be a formation of lariat structure while the introns have been removed. And what are the different enzymes or the proteins which are associated here? Can you guess it? Like what are these U1, U6, U6, U2, U1? Are they proteins? Are they enzymes? Are they RNAs? Which are they? Yes, it is NERVs, small ribosomal nuclear particles, which are called as NERVs. So these are actually the one which we talked about, snRNA, small nuclear RNA. Small nuclear RNA associated with the proteins becomes the NERVs. Okay, and these are the names of it. U1, U2. Whenever you come with these terms, remember that this have the roles. NERVs have the role in what? It is in the splicing. Splicing, you know the meaning already. It is the removal of introns and joining of exons. Okay, and there is a different types called as alternate splicing and trans splicing. So what is alternate splicing? See here, the different 5' to 3' splice sites are used during the splicing, which results in the more than one mRNA in a single pre-mRNA. If this is a single pre-mRNA, if it is exons, if it is introns, exons, introns, and exons, there will be different splice site taking place at a different times, because of which more than one type of mature mRNA can be produced. So that is what the alternate splicing means. Okay, and next, what is trans splicing? It is found majorly in the trypanosomes, where two different mRNAs, if I take it as one mRNA 1 and this is mRNA 2, they are spliced together and later they have been joined to form a different mRNA, which will definitely give the different type of protein. That is what called as trans splicing. Hope these two terms are clear to everyone. Is it clear? Is it boring? बोर लग रहा है क्या किसी को? Hope like you are able to remember whatever you have read till now. अच्छा, प्रदीप बोल रहा है yes. Okay. नो बोल रहे हो, चलो ठीक है, अच्छा है. Okay, next part. So once the processing is done, next part is export. Where export? Like from the nucleus to the cytoplasm. Okay, now this is mediated by the exon junction complex, EJC. Just remember this. Okay, and after export, tell me, like, once the mRNA are, like, if it is the sequence, if it is used for the translation process. What after translation process? Okay, from the translation process, the protein or the enzymes have been synthesized. Like, what is the next process? The proteins or like polypeptides have been synthesized. What will be the fate of this mRNA? It has to undergo degradation, right? Degradation is again by the endonucleases and the exonucleases. The combination of the endonucleases and exonucleases will degrade the mRNA. Okay, and there are again different pathways. So once you come into the MSc, like you can get a doubt. So we have learned everything from the molecular genetics right now. What is left to learn after going to the MSc? There are several things. So this is one of the things. You can also go with the 6 different pathways of how the digestion takes place, and you can, like, the one thing majorly I left about DNA repair mechanism. You can also study, like, you will be studying mostly all of this once you come into your MSc. Okay, that is left for the post graduation. And this, you can remember this risk complex. Risk, see these keywords, at least, like, what I'm trying to say is, if you do not know the process, you do not know like how it exists, at least know the keywords. If I say, okay, there is a risk complex, immediately you have to remember that, okay, risk complex, it is something which is related to the mRNA degradation. If you know this thing, that is more than sufficient to answer any type of questions that will come in your exam for sure. You have to know the term, the important terms, and where it is associated with. Nothing more, nothing less. Do not worry about like what is it, like how it is doing. Do not worry about it. But at least know, whenever I say risk, you should be able to connect with the mRNA degradation. That would be sufficient for at least for you, your level. Next, RNA editing. I told you again, this editing is like complex process. Majorly there are 2 different pathways, so you can skip it entirely. Similar to that of mRNA splicing, the splicing which we have talked in case of mRNA, there is also called as protein splicing, where the intines are removed and the extins are kept. Okay, this occurs in the protein, the polypeptide which is formed after the mRNA. Even in this, some parts are cut, some parts are remained and joined. That is called as protein splicing. Do not think that splicing only occurs in the mRNA. It can also occur in case of proteins, which the terms are called as intines and extins. Okay, so this completes the process of transcription. Although I did not go in detail about the initiation process, elongation process, termination process, but I told you about the termination, like how exactly the termination occurs in case of prokaryotes and eukaryotes in the transcription process. So at this very last moment, so whatever I have said, if you are able to recall at least 90 to 95%, na, you will definitely able to attend most of the questions from the molecular genetics. That you will that you will tell me after completing your exam, that's for sure. So this is the confidence that you should have, that molecular genetics is not complicated. It's simply that such that you have to recall and revise whatever you have learnt. That's it. Tracking of genetic code. So next, for our understanding, once the mRNA sequences have been transcribed from the DNA, the next part was translation. So before translation to occur, we know that it is the amino acids which have been synthesized, but how exactly the amino acids have been synthesized, how the coding dictionary works, that was cracked. That was called as cracking of genetic code. And it was for the first time, it was by Gemo who suggested that codon have like 3 base pairs, but he made this correct. But what was wrong? The codons are overlapping. What does it mean? If I write it as AUG, GUG, he meant to say that these 3 bases will give us the one amino acids, the next 3 bases will give us the next amino acid, and the next 3 bases will give us the next amino acids. So is it true that this is overlapping? The complete coding dictionary is overlapping? No, it is non-overlapping, right? So that's what one thing was right, one thing was wrong. And in the non-overlapping system, each base of mRNA occurs only in the one codon. That means if this A is present in only amino acid 1 and G is present in only amino acid 2, this do not overlap with each other. Okay, that's what it means. In the non-overlapping, if I have to show it clearly, it is the sequence is this. The first 3 will specifically give rise to one amino acids, and the next 3 will give it to the second amino acids. There is no overlapping between the codons, or there are no overlapping between the nitrogen bases of the one with the other. That's what the non-overlapping means. And it was in the with the genetic study of T4 phase. Again, T4 phase came here. Where exactly we studied about T4 phase? In which experiment earlier? If you can recall and tell me. Yes, we studied it in the previous class. For which experiment we studied the T4 bacteriophage? Tell me about T4 and T2 both. Which experiments we used? What about T2 bacteriophage? Benzar experiment T4. Okay, Archie and Chase, good. So T2, remember, this is how you have to recall. Every time you read something, you have to go back and revise whatever you have learned before. So T4 bacteriophage that we discussed in the Benzar experiment, and the T2 bacteriophage, it was for the Hershey and Chase experiment that we used to discover DNA is the genetic material. Someone is T2 phase, why you went for the Michelson? See, students are getting confused here. See, T2 phase, Michelson and Stahl experiment. What was the experiment is all about? What is the experiment is about? N14 and N15 DNA, right? Okay. So is there any relation with the T2 bacteriophage or T4 bacteriophage here? So do not get confused with the things. This is not the one which you are talking about. We are talking about this two. These two are the experiment. If you are getting confused, please go and check the PDF once after completing this video. So you will get to understand it. Coming back with the same T4 bacteriophage studies, it was concluded that it was by again Crick. Crick, what are the other contribution? If I ask you collectively, what are the contribution of the Crick? Will you able to tell me? Can you list it out? What are the other contribution that we have listed till now? What are the other contribution of Francis Crick? One is related to the genetic code that we have seen here. Other one is related to central dogma. And the third one is related to what? It was DNA structure, double helical structure. These were the contribution of this one. And also like one more thing, very good. We talked about the Wobble, like we will be talking about the Wobble hypothesis. Good, good. Colinearity, fine, yes. See the contribution of the Crick. You have to remember all of this. Do not get confused. This is how you have to prepare. See, whenever you get a scientist, you list all its contribution at the one place so that you won't forget forever. Colinearity and also central dogma was given by him. Okay, these are two different things. Central dogma talks about like the information passes from DNA to RNA and to protein. That was a different thing. Colinearity, it is saying that whatever basis that we see in case of DNA, that will exactly come in case of the protein structure, which is in the codon dictionary. Both actually is collinear with each other. Whatever, see, if there are any changes that we make in the DNA, if something is deleted, that will be also deleted in the amino acid sequence. Something is added, that will be also added in the amino acid sequence. That is what exactly it means. Okay. So remember all of this. These are all the contribution from the Crick. And based on these studies, it was known that codon is non-overlapping and it is degenerate, commaless. And what is degenerate? Single amino acid can be coded by more than one codon. Codon 1, codon 2, codon 3 and many. So that's what the degeneracy, the property of degeneracy means. And next, this was the second type of study. First study from the GEMO, like I'm going with the only important one. The first study from the GEMO, it was known that the codon consists of 3 bases. The second study showed that it is non-overlapping, degenerate, commaless. Third study from Nirenberg and Enrich Mathy, the 3 different codon dictionary was deduced, where like UUU codes for the phenyl alanine, AAA codes for the lysine, and triple C codes for the proline. Then what about triple G then? Why it was missing? What does it codes for? Yes, quick. So what does this code for? Triple G. Yes, remember it does not have any amino acid here. Okay, remember it. So next, OCHA and co-workers and the corona, they collectively decoded all the 64, all the rest of the 64 codons. Total of 64 codons, 3 were already deduced. So next, after that, whatever was remaining, that was deduced by the OCHA, co-workers and corona. Corona and OCHA. Remember these two important scientists' name. Someone is saying glycine. Glycine is coded by triple G. Seriously, you are confused or I am confused? Okay, triple G codes for glycine. Satvika said nothing. Others are saying glycine. Tell me quickly, like, is it yes? Okay, if you're confident, tell me yes. Glycine is the one which you are saying for triple G, right? The answer that you people are giving, but okay. So let's verify it. You're confident about this. Am I missing something? So let's check it once. Okay, I'll do it after this class. Fine. Next, there is 3 different codons, which is called as stop codons. That was given by this Greeks. Remember the Greeks term also. So here the Greeks terms are very important. From every other year you will get this, the 3 different codons which are like called as stop codons, which do not code for anything, which is like UAA and UGA and UAG. Okay, remember this. UAA, the Greek term is like ochre. UGA, it is opal, and the UAG, it is amber. Remember this 3 Greek terms for the 3 different stop codons, which is very, very important. Okay. The next, except for leucine, serine and arginine, the first 2 letters read from the 5' end are common in the codons of a single amino acids. So what is it? What does it mean? I'll tell you in the next slide. Now, properties. So this is again the most important one which you have to take care of. Most of the things I have already told you. It's triplet, that is it is in the 3 base codons, degenerate, unambiguous, and it is like nearly universal, not completely universal. It is commaless, non-overlapping, and contains a certain specific start and stop codon. Stop codons we have listed out. What are them? That is like UAG, UAA and UGA. But what about the start codon? It is like mainly AUG and sometimes it is GUG. It is in case of prokaryotes, but in case of eukaryote, it is strictly AUG only. Okay, so these are the properties. The degeneracy, no need to remember this, but have one reading of it. That one codon, only one codon is present for methionine and tryptophan. 2 codons are present for the 9 amino acids. 3 codons are present for the isoleucine, the only one, and the 4 codons for the 5 different amino acids which are listed here. 6 codons for the 3 different amino acids. Even I do not remember this. So just look at it. Nothing to be remembered here. Okay. So next comes the another important part, which is regarding the degeneracy. See why this degeneracy is occurring. What does degeneracy means? Like single amino acid, it is coded by several codon, different codon, not by just by single codon. several codon. What is the possible reason for this? There are two possible reasons. Because of presence of more than one variety of tRNA, each with a different anticodon, which will recognize the same amino acid. If, for example, I am talking about isoleucine, isoleucine is the amino acids. So this amino acid is recognized by the tRNA, which has the anticodon at the top, anticodon at the bottom. Okay, so this have the multiple. This is one. This can be like second different type of tRNA which has a different anticodon, but still it can only recognize the isoleucine and bring up the isoleucine amino acids on its top. Because of this presence of the several tRNAs for the same amino acids, degeneracy is occurring. And next, it is the ability of the anticodon on a tRNA to base pair with more than one codon. That means if this is the amino acid, let's assume that it is AU and let's assume this is G. Okay, this is G or C. Third, 1, 2, 3. This is in case of mRNA I am saying about. I am talking about the mRNA. When the tRNA comes, let's assume that tRNA is coming here. So here this will have what? It will have U, A, and either in this place if it is having U or C. Just a minute. Let's assume it is AUG. So the tRNA which is having U can also come and pair with this, and the tRNA which is having UAG can also come and pair with this. There are two different tRNAs which can come and bind to the same amino acids with having two different anticodons, and there is no strict base pairing between the last, because G will pair with C, right? G will actually pair with C, but instead of if it is U, or even if it is U, it can also come and pair. And this particular property where the last part, the first in the anticodon and the last in the codon, last in the mRNA codon. So this will not follow the strict base pairing, where there is some compromise which is called as Wobble hypothesis. This is explained by the Wobble hypothesis given by Crick. Now, how to remember this table? This table is important. How to remember it? I'll tell you. One simple trick. See AC, see the AC. AC is always comfortable. There is no problem in the AC. Like whenever you are in the AC. So remember that when there is AC, that for C there is no compromise. Always base pairs with G. A will always base pairs with U. There is no compromise in this AC. But where there is a problem, there is a problem with our UG. Okay, there is a problem with our UG where we do not get sufficient time to prepare for this ICAR PG exam. So that's the problem. The problem is always with the UG. Remember that there is a problem with the UG where there is no strict base pairing that is taking place. But how to remember that, whether it is U, whether it is C, and how to do it? I'll tell you the trick for this also. It's very simple. Now, G is what? It is purine or pyrimidine? It is purine, right? G is purine. So purine will always pair with what? Pyrimidine. Agree? So G, what is its base pairing property? It generally base pairs with C, which is obviously the pyrimidine. So apart from pyrimidine, like apart from C, which other pyrimidine is left? It is only the U that is left. So it has to obviously go for U. Since it is like we are talking about mRNA, T will not come into the play here. So that's how you can remember. It is either C or U for G. Next for U, what? Like urine, U is what? Pyrimidine. The pyrimidine will always base pair with what? Purine. So what is its normal base pairing? It is with A. Apart from A, what is left? G, right? This is the another purine. So that's how like it is A or G. And next we have inosine. Inosine, there is no trick. Just remember that all two from pyrimidine and one from purine. So A, U, C. So that is what the inosine will pair with. So you have to remember from this Wobble hypothesis. It is very, very important for you people. Okay, the exception, again there is some exception for stop codons also. Usually I told you the stop codons which are UAA, UGA and UAG, it will not code for any of the amino acids. So it will be like a signal to stop or terminate the process of translation. But there is an exception where it is recognized as a 21st amino acid, which is selenocysteine, which is coded by UGA, and there is 22nd which is pyrrolysine, which is coded by UAG, but it is not universal. It is not found all across all the organisms. It is very specific and rare, but the exceptions are true. That's it. That's what you have to remember. Next, now comes the actual process of the translation. So in case of translation, what exactly the translation is? It is like mRNA to the production of amino acid sequence, that we know it. So now how it is different from prokaryotes and eukaryotes? Let's talk about the mRNA. mRNA, there is a single mRNA which can code for more than one polypeptide. What do we call this as? It is called as polycystonic. And I told you in the eukaryotic mRNA, they are monocystonic. That means if I have one mRNA, that will only give us the one polypeptide, not more than that. But in case of prokaryotes, one mRNA will give us the more than one polypeptide. Okay, because it has a genes for more than one, like it has a genes more than for one. Example is lac operon, you can take. Lac has a single promoter operator, but genes were three: lac Z, lac Y, and lac A, right? ZYA. There are three genes which are present in the single. That's how you can remember this part. Okay. And regarding the short life, the mRNA are short-lived in case of prokaryotes, but comparatively the half-life is higher in case of eukaryotes. And regarding the ribosomal sites, they have multiple ribosomal binding sites, but it has the single ribosomal binding site in case of eukaryotes. I'll come to that later. Now, differences with respect to the initiation, which you have to mostly remember. When again, remember, for replication, for transcription, and for translation, there are three fundamental processes, which is like initiation, elongation, and the termination. This is common to all. We have to understand like how the process is initiated, how the elongation takes place, and how the termination takes place before the replication, transcription, and the translation. Okay. So now translation initiation in case of prokaryotes, it is, yes, it is AUG, but the GUG and UUG will can also act as a initiation codon. So while initiating the process, what is the amino acid which is synthesized? Methionine. So now some question was asked in the one of the, I think, mock test regarding the AUG codes for the example for the universality. Codons or genetic code is nearly universal because of the reason that AUG will not always code for methionine. AUG will sometimes, or in the prokaryotes, it will code for formyl methionine. Is it the true example for the universality? It is a true example why we are saying that the genetic code is nearly universal, not completely universal. Think again and tell me, like, is it the real example? Tell me one thing, the formyl methionine, as it is, formyl methionine has been synthesized from the process of mRNA, or the methionine is first synthesized and later the formulation takes place. Which one is true? Whenever the AUG is coded, whether the methionine first comes and then the formulation takes place, or formyl methionine itself comes. So it is like the formulation takes place after the process of, or after the production of methionine, right? So how comes like we can say that it is not universal because of this reason? This is not the example. Here AUG always codes for methionine, but in the prokaryotes it undergoes formylation. That's it. And GUG, it can be the example for the near universal, because GUG codes for, generally it codes for valine. Okay, but when it is a start codon, it will codes for the methionine, which formulation takes place after that. And also for UUG, it will initially act as the formyl or the methionine. Okay, so that's what you have to remember. But in case of eukaryotes, there is a strict behavior that AUG always codes for methionine. No other codon can no other codon can replace it. And AUG in case of eukaryotes. What about the archaea? Archaea resembles with the prokaryotes or eukaryotes in this character? It resembles with which case? So the archaea resembles with the eukaryotes, where there is no formal methionine. It is like simple methionine that is acting as a start codon from the codon, or this methionine is the first amino acid which is synthesized from the codon of the AUG. That's what you have to remember from the initiation of transition. So now the next thing, how does the structure look like? So here you can see there are start codon, stop codon, start codon, stop codon, and intercystonic regions. and before this two there are certain regions called as leader sequence and the trailer sequence. This is with respect to the prokaryotic mRNA, and in case of eukaryotic mRNA, the start, the stop, and the poly A tail because of the capping and tailing, which you don't see it in the prokaryotes. And there is two different regions called as UTR, untranslated region, 5' untranslated region, 3' untranslated region. They are nothing but the leader and the trailer sequences. Okay, now translation initiation in case of prokaryotes. What are the transition sequence which are responsible for the translation initiation in case of prokaryotes? If you remember, so it is some transition sequence which are present here, which is called as what? Shine dull Carnot sequence, which you have to remember. The Shine dull Carnot sequence, very good. Okay, students are answering it, very good. So this is called as Shine dull Carnot sequence. What about the eukaryotes? Similarly, we have similar sequences in the eukaryotes, although like it is not present in all the cells, but still there is something. Yes, very good. It is called as Kozak sequence. Yes, good. So if you want to see the figure, this is how it looks like. There is some difference between the Shine dull Carnot and Kozak. Shine dull Carnot, they are actually present. This is the Shine dull Carnot sequence, which you can see here, where the 16S RNA, the ribosomes of 16S RNA, the smaller subunit or the larger subunit. It is the smaller subunit which comes and binds to this place first, and here you can see this is the start codon. So the binding actually occurs behind the start codon where the Shine dull Carnot sequence are present. But in the eukaryotic type, the sequence, the Kozak sequence, within the sequence itself, there is a start codon. So this is the one difference between the Kozak sequence and the Shine dull Carnot sequence. And both in the prokaryotes and eukaryotes, it is a smaller subunit of ribosomes which will have the role to go and first recognize and bind to this mRNA sequence. That you have to remember. Now, next about the tRNA. tRNA, it was again by Crick who postulated the correct alignment of amino acids in the protein is made up of adapter. He did not mention about tRNA. He told that there should be something called as adapter which will help in the correct alignment of the amino acids in the protein with the mRNA. So this was later to be known as the tRNA, and it was for the first time like existence of tRNA was demonstrated by Ugland, and this is second important thing. Third important thing, it was by Ole and the co-workers who determined the sequence of yeast alanine. The tRNA which was coding for or which was having the amino acid alanine from the yeast was decoded by the Ole. The three important points you have to remember it. Okay. Next, arms of tRNA. So there are three different arms, like 4 different arms and 3 different loops you can see with the variable arm present. So here it is D arm, diuridine, and this is thymine, and this is anticodon, and here it is like amino acid acceptor R. Okay. So now I'll ask you one question. The RNA will have the thymine or not? We know that RNA will have a uracil instead of thymine, but is it strictly universal that all RNAs will have only the uracil, not the thymine? So remember, DNA will not extensively go any kind of changes, but RNAs, since it has the catalytic role based on the condition of the cell, it will always change its structure. The structure is not like very stable. That's why we say it like unstable, and there is lot of variations that we see in the structure. In that, one good example is like conversion of the thymine. Wherever required, the thymine will be also incorporated in the structure of the tRNA. Is nothing but the RNA, right? We are talking about the RNA only. This is the RNA, type of RNA. So in the RNA, you can see the T. So it is not strictly universal that it is always the U that is present in the RNA and T is missing. T will be also coming later. It is not during the transcription process. After the transcription is done, later there is a modification of the bases that can take place, where you can see the T arm here. And this particular arm, here this arm is like anticodon arm, which goes and correctly aligns with the mRNA sequence. This will carry the amino acids based on like what codon is present here, and this is D arm and this is T arm. That's what you have to remember, and also try to like read once the complete functions of all of these arms. That is important. And variable arm, why the name is called as variable arm? The name variable arm because the length of this arm like differs from tRNA to tRNA, cell to cell. That is why it is called as variable arm. So now there is one uniqueness between the initiating tRNA and the rest of the tRNA. So what is it? Initiating tRNA and rest of the tRNA. So what is the fundamental difference between these two in terms of structure? Yes, what is the fundamental difference between the structure? Yes, 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 very good. So there is a three unpaired nitrogen bases that you can see in case of initiating tRNA. So can you guess it, like why it is required? Why this unpairing is required in the only in the initiating tRNA, not in the other tRNAs? Why it is so, like it is required that initiating tRNA should have this unpaired three nitrogen bases at the 3' end. Remember, it is at the 3' end. It starts with 5'. See, this linear tRNA after synthesis, it is forming this clover leaf stage. What is a tertiary structure of this tRNA? It is like L-shaped stage, more packed stage. So why this three nitrogen bases needed to be unpaired here? What is the reason? Why there should be difference between the initiating tRNA and the other tRNA? It is because of the reason that we have three different sites in the ribosomes, which is called as EPA sites. But if you remember, in the first tRNA, which will come to this particular structure, it has to land on a P site. It can't go to the, it should not go to the A site. The first tRNA, which is like initiating tRNA, should always come and place itself to the P, and the next incoming tRNA should come and place it to the A. Because of this difference in the function, there is some structural difference which will help to come and land on the P, not on the A. And this is the whole complex which is formed where the actual process of translation takes place, which is called as PAE, EPA site. And what is to be remembered for you, like you have to compulsorily remember the prokaryotic ribosomes and eukaryotic ribosomes and its parts, where it is 70s, we all know this, but after once it is split, it is 30s and it is 50s. This is also we know it, but plus the proteins also very much important. The smaller subunit in the prokaryotes has 16s rRNA and the larger subunit has 23s RNA and 5s rRNA. Next, this is 23s rRNA. Okay, I always do not pronounce this. 23s rRNA and 5s rRNA. And in case of the smaller subunit, which is the 40s, it has 18s RNA plus 33 proteins, and this has 20s 28s rRNA, 5s rRNA and 5.s rRNA, which is most important. Okay, and in this 5s RNA is synthesized by 5s RNA, 5s rRNA. It is produced by which RNA polymerase enzyme? I told you there is a difference. Okay, rRNA produced by which enzyme? Which RNA polymerase? RNA are generally produced by RNA polymerase 1, right? But this 5s RNA, it is produced by the RNA polymerase 3. Very good. Okay, RNA polymerase 3. That's what you have to remember. Next, we talked about the initiation process, where the smaller subunit goes and attach first on a Shine Dalgarno sequence, and here it is in the Kozak sequence. And this is not that simple as I'm explaining. There are like certain steps, there are a lot of enzymes, lot of factors, lot of proteins that come step by step. Each of these proteins will have some role. So I won't be explaining in this right now because you can't really remember if you have not read it previously. Anyhow, like if you have already gone through it once or twice, most of the master course students have already done this. So you can just have a clear look at it and revise the ones, what are the roles of different enzymes. That will definitely help you. There are like rare chances that they will go in deep and ask about the different functions, but since like level of paper is rising year by year, and also like see, when we were preparing, there were no such series in the sessions in the YouTube. Like we did not have the marathon series, like we did not anything have regarding the mock test, regarding the discussions, or nothing was available. So the competition among the students is increasing. The things we knew while we are preparing, you have, you know, like a lot of more more things, like a lot of things that we have not known when while we are preparing. So level of paper can rise from year to year. So these things are like not compulsory but optional. So if you already know it, try to remember it once or twice, like revise it. Make sure like whatever you can remember, you remember for the exam. That can definitely help. So elongation. In case of elongation, there are three factors, elongation factors in case of prokaryotes. And again, this is have the three factors, like multiple factors in case of eukaryotes. That is there. This is the difference. And in the prokaryotes, it's very simple. It is Tu, Ts, and G. These are the elongation factors. And this particular tyromycin, it is actually the inhibitor of the Tu, which will stop or it will not allow the Tu elongation process to take place. Remember these examples. These examples can be directly asked in the exam. Okay, now comes the termination. How does the termination takes place? See, in case of translation, remember every step has some factors. The initiation is called as initiation factors, elongation is called as elongation factors, termination is by termination factors. Termination factors, they are called as release factors generally. Release factor 1, 2, 3. And even in the prokaryotes it is 3, and in the eukaryotes also we have 3, but the notation is like E, eukaryotic release factors, and here it is just the release factors. That difference you can remember. And again, the stop codons are same. There is no difference in the stop codons in both prokaryotes and eukaryotes. They are one and the same, and this is universal, that this will act as only these will act as a stop codon. There is no other stop codons which are present. Out of 64, 61 will code for the amino acids, and the if one codons will code for the amino acid, only 3 codons which code for do not code for anything, which are called as stop codon. Okay, next we have a term called as polyribosomes. What exactly is the polyribosome? Polyribosome, the meaning is simple, that whenever there is a translation taking place, it doesn't mean that the single mRNA is like introduced with the single ribosomal complex. Okay, at the same time, the multiple ribosomal complex can attack. It's like attacking the same mRNA and trying to get the information out of it, trying to synthesize the protein out of it, which you can clearly see here at different locations. It starts from here. For example, one starts from here and goes here, and next comes here and binds. It starts moving. Third comes here. That's how the polysome or the polyribosome means, where simultaneously a single mRNA can translate the multiple proteins at once. The energy efficient process. It is present in both prokaryotes, eukaryotes, including the chloroplast and mitochondria. Okay. Now, at the end, what exactly is important for you from the translation is knowing all of this process, plus this is most important. Know the complete table, go through the complete table, know each and every step. With respect to the mRNA, what is the difference between prokaryotes and eukaryotes? With respect to the RNA ribosomes, what is difference between the prokaryotes and eukaryotes? With respect to the coding dictionary, what is different in prokaryotes and eukaryotes? All these things, like tRNA, what is the difference between the tRNA in prokaryotes, eukaryotes? What is the difference in the process of translation, and what is the difference in the process of translation initiation? All of these are important. Okay, so we have already discussed majority of the things which are present here. Okay, we have discussed majority of things, but anything is left out, you can discuss from there. So that completes the process, the molecular genetics part 2. It should have completed in the part only one single video, but I have taken the 2 videos. In the next part, I'll be discussing about 2 important things, which is regulation part plus mutation part plus transposable elements. These 3 we can take care in the next class, hopefully by tomorrow morning or by tomorrow evening. I'll let you know the timings. Okay, how is it? Okay, good. Feeling somewhat relieved? कुछ अच्छा लग रहा है कि ओके ठीक है, मॉलिक्यूलर जेनेटिक्स कुछ तो अगर कवर हो गया, अभी एटलीस्ट 70-80% तो कवर हो गया, ज़्यादा तो कुछ बचा नहीं है. एक बार इसको पीडीएफ को पढ़ लेंगे तो हो जाएगा. फीलिंग इट और नॉट? Yeah, one thing I can see it here, like most of, like I think almost all of you are from the master course only, so that's the good thing. That's the good thing, that you are the one who are taking the benefit from whatever we are providing. And also I understand that a lot of people will also watch the recorded session after once it is live stream is ended. So no matter like what is the competition, do not worry about others, like what others are preparing, how others are preparing, how like the things are benefiting to others, what are the lacunas of yours. Do not worry about anything. Just trust the process, believe in yourself. It doesn't, see, the things will not always correlate. If you know everything, you score well. This will not always correlate. If you know few things but you know them correctly, even then you can score nice marks in the exam. That entirely depends on like how you apply your mind during the exam day. So at this last moment, even you ask like every guy, no matter like whether he or she would score rank 1, rank 2, everyone are pressurized, everyone will be in a source of tension, no matter like they have started from last 1 year, last 2 year, or they have started from last 6 months. Everyone will have the tension. That is common to each and everyone. The only difference that will make is like how you are handling it. Whether you are wasting your time searching for like what is the cutoff, how much marks should I score, so should I score this marks, I'll go to this college, I need this college, this college is fine, non-JRF is fine, JRF is fine. If you just try to calculate all of these things, try to search things in the YouTube, so what is the cutoff, like see, we also have all the cutoff data, okay, but I did not like publish or did not like spread it out at this time because this is not a right stage. Do not worrying about the cutoff, worrying about the marks, worrying about like which college should I get, like which college is good. All of these things will be discussed later. Now, even though like everything will keeps on going in your mind, but do not spend a second worrying about these things. Do not spend a second googling it, doing it on YouTube, watching some videos related to that, or watching the videos like how to prepare. Do not do such mistakes. Just focus, just consume the content, whether it is in the YouTube, whether it is from the books, whether it is from the course, mock test. Just try to recall, revise. That will be sufficient for all of you. Okay, so it was a very nice session. I feel somewhat energetic than morning. I hope like whatever I wanted to convey, I've conveyed it in a very simplest way. So if you feel that I have like simplified the molecular genetics, I want you people to comment on this video that molecular genetics now feels easy. Like molecular genetics is easy. Type the word molecular genetics is easy on the comments after the stream is ended. Since I want to see the comments, the chat will anyhow vanish after the stream is ended. I want you people to comment so that I'll also get the confidence. Okay, our students have learned something great, so they will definitely do well in their exams. Yeah, today I searched for the cutoff data. Please do not learn it. Yes, sir, but there is so much to learn. There is nothing much to learn for you people. Like whatever I said is na, like someone is doing the self study, you go and ask them. They won't be knowing like 50% of this. That's for sure. They might have mugged up. They might have tell you like, okay, this is this, this is this. But if you ask the meaning, little meaning, if you ask the mechanism, the reason behind, not everyone can tell it. Only the students who are attending these sessions, who are getting the lectures from the top rankers from the IRA and the other institutes, they will be only knowing these things. Not everyone. There is nothing much that you have to learn for this stage. Yes, like learning is unlimited. Even I have to learn so many things, but for the exams that you are giving, this is more than sufficient what I have explained. Yes, I want. Yes, Vinay is already saying that molecular genetics is easy. Please come post a comment on this video after the live stream is ended. Since I told you this chat anyhow will vanish, I won't be able to see it later. Thank you so much for watching this video. Prepare well, prepare well. Do not worry about anything. Try to attend the mock test and do not attend the mock test. All the mock tests at a stretch once. Attend only 2 to 3 full length per day and revise them and see like what is like weak areas, what are your strong areas. That will definitely help you to. And regarding general agriculture, there will be hardly 4 to 5 questions or more than like maximum max to max, including the biochemistry, biotechnology, stats, 10 questions will be asked. So if you are too much worried, if you have not started the general agriculture, do not give so much of time to that. Try to revise the major subjects like genetics, plant breeding, plant pathology, and microbiology and the seed tech. It's very simple. You can revise it in a very minimal amount of time. So please make sure to make some good strategy so that before the exam, at least 60 to 70% syllabus is covered. Do not distract yourself for the unnecessary things, okay, from the YouTube. And whatever you are consuming from the YouTube, it should be only the content which is required, not anything else, not the logs, no, nothing more, like not the success stories, not about the cutoff or nothing else. Only the content. Wherever you are getting, do it. Mostly like you will be getting it in the AgriAddict YouTube channel. So please do it to make sure to consume that content only. Put it in a 1.5x, 2x, save some time and clarify all your doubts. Okay, fine. Thank you so much for attending. See you in the next class. I'll post the timings in the YouTube and also in the WhatsApp group. So if you have not yet joined our any of the WhatsApp and the Telegram and Instagram community, simplest thing is just join the WhatsApp channel and the Telegram channel. Joining these two channels and also the following us in the Instagram and the YouTube, that helps. So if you are not able to join any of the WhatsApp groups, there are dedicated groups for the different subjects, but they might be almost full. So just simply join the WhatsApp channel and the Telegram channel. All the updates will be provided there. If you are a free learner, anyhow, if you are a student of master course, we'll be doing it in the WhatsApp master group that we have. Okay, thank you so much.